Okay, so I want to mention before we get started here that the two assignments that I was going to have be due today, obviously I'm not up at WT this morning. So what I want to do is just have you submit those assignments, the activities 2 and 3, and then lecture activity 1.1. You can just bring those to lecture next Wednesday if you attend by lecture on that day. So that's of next week, or you can submit the assignment under the correct Dropbox on WT class. Okay, again, that's under the Lessons tab, Unit 1. Okay, so we're going to start where we left off on Monday. Okay, so double-blind, placebo-controlled, randomized experiments are considered the gold standard in science. However, they can be unethical or impractical to conduct with human subjects. So some of the examples that I'm going to cover, one of them we've actually covered before, concerning birth control. It would not be ethical to conduct a study on birth control in which your controlled subjects would receive a placebo, okay, that has absolutely no impact on preventing pregnancy, okay? So it's not going to prevent pregnancy, it's a placebo, whereas the experimental group is going to receive some type of birth control, okay? Obviously, that's not ethical to conduct on human subjects because clearly those in the control study, those women would have a high likelihood of becoming pregnant during the study, okay? So clearly unethical. Another example of an unethical study to conduct that would be double-blind, placebo-controlled, and randomized would be a study on different types of cancer treatment like chemotherapy. You would not give one group a placebo for that cancer treatment because that is not going to treat their cancer. It'd be unethical for a group to think that they were going to receive cancer treatment when they're actually receiving a placebo, okay? So in those cases, you can use correlation studies to test your hypothesis. And actually, some of you did use a correlation study when writing your experiments this past Monday. Okay, with correlation studies, a model system is used. And that's where you have a non-human organism used in genetic studies. That's to help scientists understand human genes. Okay, so this can be utilized when your experimental organism shares a gene or genes, multiple genes of interest with the human. Okay, so those genes need to be shared. One common model system that is used in science is the mouse model. Okay, mice do share a lot of genes with humans, so they're a good animal system to use. And also model systems can be used when the hypothesis would raise ethical, which we actually looked at, or practical problems in human studies. Maybe it might not be practical for you to conduct that study in terms of conducting a study on humans. And some of you actually did look at that within the experiments that you wrote. Okay, so correlations are going to evaluate relationships between two variables. In that case, your independent variable versus your dependent variable. So an example on a correlation study would be based on our hypothesis that stress makes people more susceptible to catching a cold. So that's what we're going to take a look at in terms of if there is a correlation or not. So we're going to do a correlation study. And we're going to look at figure 1.10 from your textbook. Okay, so here we're looking at psychological stress levels on the x-axis. That is going to be your independent variable. Your independent variable is always on your x-axis, okay? Looking from low levels of stress all the way to high levels of stress, okay? Then on your y-axis is your dependent variable. And it's referred to as dependent because it's going to depend upon your independent variable. Okay, so the percentage of subjects with colds is going to depend upon the amount of psychological stress. Okay, so what we actually see here is a positive correlation. As you have an increase in the amount of stress level, that is going to increase the susceptibility to colds. Okay, so there is indeed a correlation, a positive correlation there. So people with higher stress levels were more likely to become infected with cold virus. Okay, so even though there is a correlation here, one thing we have to consider is their causation. Does high stress cause high cold frequency? Maybe it doesn't. Or does one of the causes of high stress also cause high cold frequency? Okay, in this case, there would be alternative hypotheses at play. Typically, people that have other types of illnesses are more susceptible to cold, so they're going to have a higher cold frequency. Also, having other illnesses can lead to high amounts of stress. So it's not that the stress is leading to high cold frequency, it's the fact that the individual has other illnesses. Another alternative hypothesis that may be in place is little exercise. Little exercise can lead to a high frequency of colds. Also, getting little exercise can lead to high amounts of stress. So here again, it's not a cause and effect relationship between high stress and high cold frequency. It's a possible cause and effect relationship between little exercise and high cold frequency. Okay, so these are some of the alternative hypotheses that could be at play. So typically a correlation study cannot eliminate all alternative hypotheses, okay? In order to eliminate at least a number of alternative hypotheses, what you want to do is look closely at your research participants, making sure that there is no differences between the groups in terms of age. Like one group you have older individuals, another group you have younger individuals. You don't want that. You want kind of an even distribution, kind of representing the population for both the control group and the experimental group. Okay. Another one would be their exposure to the cold virus. You wouldn't want one group exposed to a cold virus and the other group not, because that's going to have an impact on your results. Okay. So you want to make sure that there is the potential of there being a cause and effect relationship there with your correlation study, and you want to try to eliminate your alternative hypotheses. Again, correlation does not mean causation. We're going to take a look at some pretty bizarre correlations here that shows that correlation does not necessarily mean causation. Okay, here we're looking at ice cream consumption and murders, and it says ice cream consumption leads to murders. Just looking at the data here, you would, and over a period of time, the amount of ice cream that sold increased, along with the number of murders increased from May to July. Okay, it appears as though there's a correlation. Clearly, there's not. Okay, so again, correlation does not necessarily mean causation. Okay, these are just kind of some fun, bizarre correlations to look at. Pirate shortage caused global warming. Here we look at a period of time over which the number of pirates increased, and then also your global average temperature increased. Okay, and there's some other fun ones if you want to take a look at that on your own. Those are the 10 most bizarre correlations. Okay, you can also refer to Table 1.1 for types of correlation studies. We're not going to cover that in lecture. So really the question is, how can we better evaluate experimental data and results to determine the value and the meaning behind correlative experiments? Okay, so really we need to be able to conduct statistics on our results. Okay, so what statistics is, it's a specialized branch of mathematics that's used in evaluation of your experimental data. I'm going to cover some just very basic terminology with you first. Okay, so your sample is just a small subgroup of a population that's used in an experimental test. And then your mean is the average value of a group of measurements. We're also going to take a look at what a statistical test is. It's a mathematical formulation that helps scientists evaluate whether results of a single experiment demonstrate effect of treatment. So this would be based on just a single experiment. Okay, with your statistical test, it's going to extend your results from the sample to the entire population, because really it's the entire population that you're concerned with for whatever you're running your experiment on. Okay, so it extends results from the sample to the entire population, meaning it's representative of the entire population. So your experiment on the sample population, it can only estimate a true underlying effect of experimental treatment on the entire population. And the reason for that is the sample is always an imperfect representation, or basically a snapshot of an entire population. It's never going to be fully representative of the entire population. The only way you could have it fully representative of the entire population would be to test the entire population. And clearly that's not possible. Okay? Also, with your statistical tests, it's going to provide you the ability to calculate the likelihood that a difference between two samples reflects real underlying differences between your populations from which the samples were drawn. Okay, so that's going to be based on the number of individuals sampled and variation within samples. Okay, we're also going to talk about what it means for something to be statistically significant. And that is a set of results from which there is a low probability that the experimental groups differ simply by chance. And that means it likely represents a true difference between your two groups, if it is statistically significant. We are also going to look at a couple of factors that are going to influence statistical significance. And one of those is sampling error. And your sampling error is the random differences between the sample and the population from which it was drawn. Okay, so here we're looking at the effect of chance on experimental results. Okay, and for when you run an experiment and you collect your data, when you're looking at statistical significance, you want a fairly low sampling error. Okay, so we're going to look at a zinc experiment, and our observation is that some forms of zinc can block certain common cold viruses from entering cells lining the nasal cavity. Okay, so our hypothesis based on that would be lozenges containing zinc reduces length and severity of cold symptoms. And in this study, we have two groups, as you can see on the right here, of the figure from your textbook. We have the placebo lozenges that are given to the control group, and then the true zinc lozenges are given to the experimental group. Okay, so both groups received placebo or the actual zinc, and it was of the same amount. was given within 24 hours of development of cold symptoms. So here we're ensuring that there is some control, that you don't have any other variables that are going to impact your results. And overall, looking at the result, this was a double-blind study of 100 individuals. So you clearly have enough individuals in the study. Okay, so overall, looking at the results, the mean recovery time was 3 days shorter in the experimental group. Okay, so our next question, is there a statistically significant difference? Okay, and based on the statistics that were conducted on this experiment, there was a 1% chance the groups differed simply by chance. So that means there's a really high chance that the groups did differ, okay, due to some inherent difference between the groups, meaning whether they got zinc lozenges versus placebo. So yes, they were definitely statistically significantly different. Also, something else that can have an influence on statistical significance is your sample size, and that's the number of individuals in both the experimental and the control group. Okay, so your sample, again, is always going to be, should be a representation of the entire population. Okay, so that means everyone within that entire population. Okay, that sample is going to be a representation of that. So in this case, bigger is better. You want as large of a sample size as you can feasibly get, because you're more likely to determine or detect a true difference. Okay, so you'll see a true effect of an experimental treatment there. Okay, so bigger is better. If you have small groups, when that is the case, researchers might be misled into thinking that their treatment has an effect. And that was actually the case in the studies that we looked at on the vitamin C hypothesis and the echinacea extract hypothesis. What was shown later is the experimental samples, or the sample size, was too small. In studies that were conducted later with larger sample sizes, it later led those scientists, those researchers, to reject the hypothesis. So clearly very important to have a large sample size. So next we're going to take a look at what is wrong with this experiment. Okay, so a student wanted to study the effect of fertilizer on plant growth. She took 2 similar plants and set them on the windowsill for 2 weeks. She watered each plant the same amount, but she gave one a small dose of fertilizer with each watering. She gathered data by measuring the plant's height and counting the number of new leaves. Okay, so our first one is measurable results cannot be expected. Well, it's going to be over a 2-week period. There is going to be plant growth, so measurable results are going to be expected. So that's not the case. That's not true. It will require too many days of data collection. 2 weeks is a fairly short amount of time, so that's not unreasonable. There is no variable factor. There is the variable factor. In that case, it's the fertilizer, so that's not true. There is no control. Well, the use of 2 similar plants in the same location receiving equal amounts of water, that implies the concept of control. So that one's not true. The sample size is too small. This one is true. Clearly, 2 plants is not going to be representative of that entire plant population, so the sample size is too small. Okay, so the bottom line on statistics, looking at statistically significant results, that shows a difference between the experimental and control groups larger than difference expected due to chance variation between groups. So that means that there is most likely a true difference between the 2 groups due to the treatment. However, it does not always translate to something that's meaningful or important. It's fairly something you want to keep in mind in terms of statistical significance. It's also not a measure of accuracy of the experiment. And when you conduct statistical studies, you always are operating under the assumption that the experiments were designed and conducted correctly. Also with statistics, it evaluates sampling error, not observer error. And of course, you can only evaluate data that was collected. Okay, so we're going to look at the differences between primary sources and secondary sources, looking at evaluating your scientific information. So with the primary source, that is an article that is reporting research results. It is going to be written by researchers and then reviewed by the scientific community. Okay, and with the peer review process, we've talked about it a little bit in the past, but I want to go over it in a little bit more detail. But with peer review, it's the process whereby other scientists that are specialists in that field are going to critique the results and conclusions of the experiment, and that's going to be before the manuscript is published into an article. So that peer review process is really going to help to increase confidence in that scientific information because of that peer review process. Okay, so to kind of visualize that process, we're going to look at figure 1.16 from your textbook. Okay, so a manuscript is going to be sent to a particular journal. Okay, the journal that it gets submitted to is going to be within the area of interest of the researchers, and it's going to be based on what the experiment was over. Okay, typically for a journal, there are going to be at least 3 reviewers that will go through and review the article in its entirety. A lot of times the manuscript will be sent back to the researchers to revise the article. Maybe they weren't completely explicit in their instructions on how, or their description of how they ran the experiment in the methods section, so maybe they need to clarify a few things there. So oftentimes you'll have a revision process, then the researcher will resubmit the manuscript, and then depending on those corrections, it may be published. Okay. However, a lot of times what happens is the article is going to be rejected. Okay. Maybe it was not submitted to the correct journal, okay, for what that journal is interested in publishing. Okay, so secondary sources, it's where you bring published information to the public. And really, that's where most people receive their scientific information, unfortunately. So with secondary sources, it's where you have books, news media, and advertisements as sources of scientific information. So that would include television, radio, and the internet. And here are some examples of where people get their scientific information. And really the best source to go to is clearly your primary sources. Okay, one of the examples on the problems of looking to secondary sources for your information would be looking at, do vaccines cause autism? Okay, there's a lot of misinformation out there on this issue, and the reason it's even an issue is because of one article that was published. Okay, so I'm actually going to send you some information on that so that you can read over that on your own, and I'll also send you a video that you can watch over autism. Okay, next we're going to look at anecdotal evidence, and what that is advice based on one individual's personal experience. Has nothing to do with experimental data. Okay, it's just based on experience. Oftentimes you'll see this in the form of a celebrity testimonial. So an example on that is, after trying every procedure, facial treatment, and product I could find, I finally decided to try Proactive. I mean, I had nothing to lose except my acne, and I was definitely okay with losing that. Since then, I've seen massive changes in my face and my confidence. Okay, so that's clearly anecdotal evidence there. You definitely want to be careful with what you listen to on it. Anecdotal evidence. One good example of anecdotal evidence that you may have heard growing up is Grandpa smoked a pack a day and lived to 94. Okay, so clearly be careful on what you listen to. Okay, so how can you discern accuracy of scientific information from popular news sources? Okay, so we're going to look at the differences between information from news organizations with science writers versus entertainment tabloids. Okay, so typically with news organizations with scientific writers, journalists will look for controversy and then get one person from each side to tell their viewpoints. Okay, this is really well-rounded news. However, a lot of times this approach does not give you the weight of evidence for each side, okay, because they want that controversy to be there. Obviously, entertainment tabloids, not a place to go for scientific information. Also, the internet, you need to make sure that you're cautious, as of course anyone can post anything on the internet. And specifically over autism, there's a lot posted on the internet. As far as vaccines, does it cause autism? Make sure that you use valid internet sites when looking for information on the internet. You want to make sure that you choose sites that are maintained by reputable medical establishments, like the National Institute of Health, or the Mayo Clinic, just a couple of examples there on that. Also, you want to choose sites with nonprofit domain names like .gov, .edu. Okay, so looking at understanding science from secondary sources, we're going to skip over the deceptive drug ads. You've probably seen a lot of them on the television, on drug ads that are deceptive. Okay, so as educated information consumers, you can use your understanding of the process of science and experimental design to evaluate any scientific story. Okay, always use the process of science and understanding experimental design and what is conducted correctly in terms of an experiment to basically evaluate that story. There are some questions that you can ask yourself to evaluate science news reportings. You can find that on in your textbook, table 1.2. And I'll just go over a couple of them. So what is the basis of the story? Your preferred answer would be hypothesis test. What can raise a red flag is if it's an untested assertion, and then also no data to support the claims in the article. Also, another one is what is the affiliation of the scientist? And when you take a look at that article that I'm going to send you over autism, you are going to discover something there that is of concern. Okay, so the preferred answer is independent, university or government agency. What should raise a red flag is employed by an industry or advocacy group, and then also data and conclusions could be biased. Okay, so you can look over those on your own. So going back to our question previously, is there a cure for the common cold? Of course, there's no cure yet, but there are prevention methods that are known. Best advice, wash your hands. With that, the rates of common cold infections are about 20 to 30% lower in populations who employ effective hand washing procedures versus those who do not. Okay, so we're going to move on to Chapter 2, where we're going to look at water and what makes it a good solvent. We're going to look at the structure of water. We're also going to delve a little bit into biochemistry and take a look at the cell itself. Okay, so some questions that are raised in the chapter are, can dead humans come back as zombies? Yes, some of these are ridiculous questions, but it serves a purpose. Okay. Another question is do sugary drinks cause hyperactivity in children? And does eating turkey cause drowsiness? So first looking at pseudoscience. It's a dubious claim that's going to be strengthened through use of false or weakly supported assertions of scientific evidence. Okay, so some things you want to look out for to identify if something may be pseudoscientific is it has the use of psychobabble. Those are words that sound scientific, they sound professional, but are used incorrectly or are used in a misleading manner. Also, there's a substantial reliance on anecdotal evidence, which we looked at the problem with that. Also, extraordinary claims in the absence of extraordinary evidence. Also, the claims it claims which cannot be proven has claims which cannot be proven false. Claims that that counter established scientific fact. There's an absence of adequate peer review, and the claims are repeated despite being refuted. So let's just assume zombies exist. Okay, zombies are not alive, but they share many characteristics seen in other living organisms. So we need to take a look at what defines life. Okay, so we're going to look at some of the attributes of living organisms. All living organisms contain a common set of biological molecules. All living organisms are composed of cells. They also have the ability to maintain homeostasis. And then most living organisms grow. They move. They have the ability to reproduce. An exception on this one you may be wondering about. The exception would be a mule. A mule is a cross between a male donkey and a female horse. Mules are sterile, so they're not able to reproduce. Also, most living organisms respond to the external environmental stimuli. And they have the ability to metabolize. So first we're going to take a look at metabolism, because it's something we're going to discuss off and on throughout the semester. So those are all the chemical processes that occur in the cell. You can have a breakdown of substances for the purpose of energy production. In that case, it would be what we call catabolism. You can also have the synthesis of substances necessary for life. So that would be to build a larger substance from smaller building blocks, which we refer to as anabolism. And then also falling in within metabolism would be the excretion of wastes generated by these processes. So we're going to go back to our example of the zombie, or zombies living based on these characteristics. Okay, can they move? Yes, they can. Can they reproduce? Well, that's kind of an interesting question there. Yes, they can reproduce in the sense that if they bite another human, that human turns into a zombie. They've reproduced another zombie, or they've produced another zombie, but that's obviously not the type of reproduction we're talking about. There is no passing of genetic information to the offspring, okay, to the human that becomes the zombie, okay? So obviously reproduction In those terms, no. Can they respond to stimuli? Yes, if a human is rummaging around, they hear that and they run to the next meal. It's like ringing the dinner bell. So yes, they can respond to the stimuli. Do they grow? Definitely not. Okay, so we're also going to look at what homeostasis is. So that's a maintenance of a constant internal environment despite a constant changing external environment. And a good example that we'll look at is maintaining body temperature. There are, in the human in particular, there are a lot of different mechanisms, homeostatic mechanisms in place to maintain body temperature. For example, if you go outside on a cold day, you'll begin to shiver. That'll help you generate heat to raise your body temperature back up to that typical 98.6. I will mention on that particular body temperature, that's an average. Okay, some people's normal body temperature may be in the range of 97, or some people, their body temperature may be at normal at 99. Okay, so again, 98.6 is just an average. Okay, so when you're exercising out on a hot day, you begin to sweat. That sweat carries heat away from it, carries heat away with it. As it evaporates, we call it evaporative cooling. So it'll bring your body temperature back down to normal levels to maintain that normal, constant 98.6 temperature. Okay, so those are some homeostatic mechanisms that are in place. Okay, it does require complex feedback mechanisms between multiple sensory and physiological systems. Okay, so here in our figure, we have the black-capped chickadee. It has a core body temperature of about 107 degrees Fahrenheit. It's actually able to maintain that body temperature even in very cold environments. Okay, so it has a lot of homeostatic mechanisms in place to be able to maintain that body temperature. Also, it's important to note that populations of living organisms can evolve over time. And so what that is a change in average characteristics over time. Okay, so we're going to look at the properties of water next, and our questionable claim is that drinking water during an exam helps students perform better. We're actually going to take a look at a study conducted by the University of East London. Okay, and that's where you compare, here's our x-axis, that would be the independent variable, y-axis, dependent variable. So the average exam score is going to depend upon whether the student brought water to the exam. Okay, so here we're looking at the group that did bring water to the exam. They did score higher than the group that did not bring water to the exam. Their average was somewhat close to 90, whereas the students who did not bring water to the exam scored a little bit below 70. So somewhere around about a 20-point difference. Okay, and this is really for the purpose of looking at the study for the purpose of looking at the properties of water. Okay, so we'll delve back into this study a little bit later. Okay, so first we need to define what an element is. Elements are fundamental forms of matter. We have 92 natural elemental atoms. They have been described by chemists and several more that have been created in laboratories. Elements are composed of atoms that cannot be broken down by normal physical means. And when looking at living organisms, our four most common elements are carbon, hydrogen, oxygen, and nitrogen. One easy way to remember this is CHON. C-H-O-N. Of course, standing for the letter of each, the first letter of each element. Make sure you remember that, because you'll need to know carbon, hydrogen, oxygen, nitrogen. Again, those four most common elements in living organisms. Okay, we also need to define what the atom is. So atoms are the smallest units that have properties of any given element. And they are going to be composed of what we refer to as subatomic particles. So within that, you're going to have protons. They have a positive electrical charge. And the way that you can remember the charge on protons, P positive, protons have a positive charge. You can also write a little plus sign here next to protons. Then your neutrons are uncharged or neutral. So you can write a little 0 next to the neutrons here. And then also they're composed of electrons. Okay, electrons have a negative electrical charge. So you can put in a little negative sign next to the electrons. Okay, also atoms have a nucleus that's going to be composed of protons and neutrons. So that positive charge and the neutral charge is going to be within the nucleus. And then your electrons are going to be located outside of the nucleus in what we refer to as the electron cloud. And remember, those electrons are negatively charged, unlike charges attract to one another. So that negatively charged electron is going to be attracted to the positively charged nucleus. So that's what prevents the electrons from just flying off away from the nucleus of the atom. Okay, it basically holds the electrons in their correct orbital within that electron cloud. Also, atoms are neutral. That's because of an equal number of protons and electrons. So you have an equal number of positive and negative charge, which ultimately means that that atom is neutral. However, they can also be electrically charged, and we refer to those atoms as ions. And so in that case, you're going to have an unequal number of protons and electrons. Okay, so next we're going to look at the relative size of protons and neutrons versus your electrons. This is kind of a good example to use in terms of mass, the mass of the proton or neutron. Protons and neutrons are about the same size, and we're going to compare it against the electron. So relative to one another, you can think of the size of the electron as about the size of a lifesaver versus the proton or neutron at about the size of a 15-pound bowling ball. Okay, so an electron is about 1/2000th the mass of a proton. Okay, so large difference in size, clearly. Okay, even though electrons are small, they're going to determine the chemical behavior of the atom, and you'll definitely see that as we progress through this on that. Okay, and those electrons, they're the parts of the atoms that come close enough to each other to be able to interact with one another. Okay, so first, one of our important elements for living organisms is oxygen. And we're going to look at what it's composed of. Here you can see the nucleus of the atom composed of the positive and neutral charge. Okay, and if you notice, the positive charge here is lumped together. Okay, so what you might be thinking, well, two positive charges are going to repel one another because they're like charges repel one another. And so what the neutrons actually do is it basically kind of operates like, you can think of it like glue, basically holds those positive charges together. Okay, so and I want you to remind you that, remember, for this oxygen atom here that we're looking at, atoms, unless we're talking about ions, atoms are going to be neutral in their charge. Okay, so we have to look at an equal distribution of negative and positive charge. Okay, so here we see our first orbital. It can only contain up to 2 electrons. Our next orbital outside of that can contain a total of 8 electrons. Okay, electrons have a tendency to pair up. As you can see, they've paired up here in that first orbital. In the second orbital, you have only 6 electrons. So at least 2 of them have paired up. So you have 1, 2 here, 3, 4, and then 5, 6. So what that means is that the oxygen atom can form up to 2 bonds with other atoms. Okay, here they will pair up their electrons. We'll talk about that here in a little bit more detail later. Okay, so overall, looking at the charge, you have 8 negative, and a charge of 8 negative, and then a positive charge of +8. Okay, so 8 protons, 8 neutrons in the nucleus, and then 8 electrons in the electron orbitals. Okay, so that there is balanced. Okay, next we're going to look at the carbon atom. It is made up of electrons, protons, and neutrons. Okay, and you can determine the number of protons and neutrons just by looking at the number of electrons. Okay, so we have 6 total electrons, which means we have 6 protons and 6 neutrons for that element to be neutral. Okay, so same number of positive charge versus negative charge. Okay, as you can see here in the first orbital, it is full. It can contain up to 2 electrons, which it does. In the outer orbital, remember we said that there can be 8 total electrons. Okay, so here we only have 4, which means that you can have 4 other atoms that are able to bond to this atom. So carbon can form up to 4 bonds with other atoms. Then our hydrogen atom, in the nucleus, as you can see, there's only 1 proton. There's no neutrons. There's no need for a neutron because you're not needing to hold the center of the atom together because it's only made up of 1 positive charge. Okay, and of course we only have 1 electron. We've got 1 negative charge, which makes that atom electrically neutral. Okay, and I will show you the hydrogen ion. It has lost its electron, so it's going to have a positive charge. And that's within the atomic nucleus. Okay, so I do want to look at the structure of water, because it's very important in the process of life. Chemical formula for water, which I am all sure that you all know, is H2O. So that means it's composed of hydrogen atoms. And it's also composed of an oxygen atom. For every 2 hydrogen atoms that you have, there's going to be 1 oxygen atom in water. Okay, so that sub 2 means that there are 2 hydrogen atoms. Here, there's nothing written. That's understood just to mean B1, so 1 oxygen atom. Also, you need to understand that atoms are always going to be joined by chemical bonds. And water is considered what we refer to as a good solvent. It's one of its distinguishing characteristics. So it has the ability to dissolve a wide variety of substances. So what a solvent is, it's a substance such as water that a solute is dissolved into to make a solution. So your solvent is what does the dissolving. So the solvent does the dissolving, And then the solute, is a substance that is dissolved in a solution. So the solute is what's dissolved, the solvent is what does the dissolving. Also, water is a good solvent because it is a polar molecule. And that describes a molecule with regions having different charges. Water is polar, thus it's a good solvent. And so in this case, like is going to dissolve like. So polar molecules goes into solution with water because water is also polar. So like dissolves like. Polar substances will have the ability to dissolve other polar substances. So water's polarity, it arises because oxygen is actually more attractive to electrons than other atoms are. So oxygen has the tendency to pull electrons towards itself. It's what we refer to as electronegative. So that's the tendency to attract electrons to form a chemical bond. Remember, it has 2 unpaired electrons, meaning it can bond with up to 2 other atoms, which we'll see in this case. To make water, it is hydrogen. So electrons in water molecules are going to spend more time near the nucleus of the oxygen atom than near the nuclei of the hydrogen atoms. So the oxygen atom is pulling the electrons towards itself. So you have more negatively charged electrons near the oxygen, which means oxygen in water is going to carry a partial negative charge. So in those chemical bonds, if oxygen is pulling those electrons towards itself, it means it's pulling those electrons away. It's pulling that negative charge away from the hydrogen. Okay, so in that case, hydrogen is going to develop a partial positive charge. So looking at figure 2.4 from your textbook, here we have the water molecule, oxygen in the red, hydrogen in the white. And if you notice, it's representing the size of the atom based on the size here that it's illustrating. Okay, so remember, oxygen has 2 unpaired electrons. Okay, so it could be either this electron or this electron that belongs to oxygen. We don't know which one. It doesn't really matter. Okay. Then the hydrogen atom has 1 unpaired electron. So these 2 electrons can pair up, creating a bond here. Here in this case, it would be a covalent bond that we'll talk about later. But it forms this bond. These electrons pair up, okay, forming this chemical bond between oxygen and hydrogen. Okay, so oxygen is more electronegative, meaning it has a tendency to pull electrons towards itself. So it's actually pulling those electrons somewhat away from the hydrogen atom. It's not pulling them away completely. We're not talking about formation of ions here. It's just partially pulling them towards itself. Okay, so that means that hydrogen is going to develop a partial positive charge. Okay, that's represented by the Greek lowercase letter delta and a plus sign. So that means partial positive charge. This hydrogen atom is also going to develop a partial positive charge here, and this oxygen atom is going to develop a partial negative charge here because it's pulling negative electrons towards itself. Okay, so that means that that molecule is polar. It has developed poles. So what water molecules do is they orient themselves so that the hydrogen atom of one molecule is going to be near the oxygen atom of another molecule due to that polarity. Okay, that's going to lead to what we refer to as a hydrogen bond. And the hydrogen bond is fairly weak. So you have a weak attraction that is formed in water between the hydrogen atoms and oxygen atoms that would be in adjacent molecules. Okay, it also can be intermolecular. It can be between different molecules, which is the case with water. And we're going to look at figure 2.5a through b in your textbook on those hydrogen bonds. Okay, first at the top, we're going to look at the bonds between two water molecules. So here we have two water molecules. The partial negative charge, remember, opposite charges attract one another. So a negative charge is going to attract a positive charge is going to attract a negative charge, okay? So the partial negative charge, remember, oxygen is more electronegative. It pulls electrons towards itself from the hydrogen atoms, which gives that partial negative charge. And then here, those hydrogen atoms of the next molecule are going to come into close proximity to that negative charge of the oxygen, and it's going to create what I refer to as a hydrogen bond. Okay, so you can see here this network that gets formed with water molecules, where this slight positive charge, you're going to have a hydrogen bond between the hydrogen here with the partially negative oxygen here. Okay, so partial negatives are going to bond with partial positives here and form this hydrogen bond. Okay, this is what explains surface tension of water. If you've ever placed a piece of hair or something that's not very massive, you place it on the top of water, it does not fall into the water, okay, because of that surface tension present there. Okay, so hydrogen bonds, when you're looking at them in low quantity, are fairly weak. However, if you're looking at them in large quantity, like you would be if you had a bowl of water, it's going to have a high surface tension. Together, those bonds are going to be strong. Okay, so water can dissolve other polar molecules. And polar molecules are what we refer to as hydrophilic. Hydro meaning water, philic mean loving. So that means water loving. So hydrophilic substances are going to readily dissolve in water. So remember I said like dissolves like. Water is a polar molecule. Water is going to dissolve other polar molecules. And then nonpolar molecules, those are atoms of molecules that do not carry any partial charge. So they're not partially charged. And so those are nonpolar. They don't create any poles within the molecule. Remember, like dissolves like, unlike is going to repel. So nonpolar molecules will not dissolve readily in water. So water is not really going to dissolve nonpolar substances. We refer to that as hydrophobic. Hydro meaning water, phobic meaning fearing, so water fearing. And an example on that would be oil. Oil and water do not mix. Okay, so on the next figure, figure 2.6 from your textbook, we're going to look at water as a polar substance. Okay, so here we're looking at dissolving salt, which is made up of sodium atom and a chlorine atom, sodium chloride. And here we're looking at water being added. So we're adding together water and salt, and we're looking at the salt, how it dissolves in the water. Okay, so both are polar substances. In this case, you're actually looking at ions. Sodium has a positive charge. The chlorine atom has a negative charge. So we have a chloride ion, sodium ion. And then water, remember, it has a partial negative charge, hydrogen has a partial positive charge. So what happens when you add sodium chloride to water is basically the negative charge on, and we'll look at this here first, the negative charge on the oxygen atom is going to attract the positive charge on within sodium chloride. Okay, so it basically pulls away that sodium atom completely away from the chlorine atom. And so the negative charge is going to surround that positive charge. Then that slightly positive charge of the hydrogen atoms are going to surround that negative charge of the chloride ion. Okay, so that's how water is able to dissolve salt, is it pulls those atoms away from one another and completely surrounds them, thus dissolving the substance. Okay, also water is going to facilitate chemical reactions. And that's because it's such a powerful solvent. And with chemical reactions, those are changes in chemical composition of substances. Now, a reactant is the starting material in a chemical reaction. And so you can consider solutes in a mixture as reactants. Okay, and then your product is a modified chemical that results from a chemical reaction. Okay, water also has the ability to moderate temperature. And what I mean by moderate is basically it makes it less extreme. So when you add heat energy to water, what it does is it disrupts the hydrogen bonding between the water molecules. So it's disrupting the bonding between the hydrogen and the oxygen between water molecules. And so that heat energy can be absorbed. And that's going to be without changing the temperature of the water. Because that heat energy can be absorbed, the temperature of the water is not going to change. Now, it's only after the hydrogen bonds have been completely broken that you can add heat to increase the temperature of the water. Okay, so that initial input of energy is going to be absorbed. Okay, so going back to our questionable claim, drinking water during an exam helps students perform better. Really, that hypothesis requires more substantiation. Okay, so with our properties of water, yes, water it does help to regulate blood pressure. It does have the ability to deliver dissolved nutrients to the body, including the brain. We do know that water helps to maintain body temperature at that about 98.6 degrees Fahrenheit. Okay, so is this really clear evidence when considering that? Well, yes, those properties that we looked at, they might help to deliver nutrients to a harder working brain, and this might increase performance on a very stressful exam. But are there really other reasons that could be responsible for students doing well in an exam? Maybe the students were more prepared. The ones who brought water to the exam happened to be more prepared. So in that case, it was the preparation that would explain the reason for those students doing well in the exam, and not the fact that they brought water. Okay, you might consider those who are well prepared are going to bring water with them wherever they are, and they're going to be prepared for the exam, so they're going to do better on the exam. Okay, what the study actually indicated was that the students who brought water were actually upperclassmen. They were more experienced test takers and more relaxed, and were able to perform better. So a good scientist will always evaluate alternative hypotheses. You always want rigorous testing of your alternative hypotheses. That's really the major difference between bad science, excuse me, having a hard time talking today, unsubstantiated pseudoscience, and then good science. Okay, and then the good scientists will not perform only experiments that will help to confirm their hypothesis. They want to get to the real answer, so they will seek out ways to falsify their hypothesis. So in that case, they'll develop and test alternative hypotheses. So other tests will be conducted. Okay. Let's take a look here. This is actually a good place to stop. We will stop here. And don't forget that Monday is Labor Day, so we do not have class on Monday. We will start back up on next Wednesday. I hope you all have a nice break.